My name is Raphael Park. I'm a PI at Scripps Research. Before I start, I want to apologize, everyone. I have a terrible sore throat. Usually, my voice is much more beautiful than what I am right now, but hopefully, I can get through this presentation. If you don't hear me well, or if you don't understand anything I'm saying because of my voice, uh, please feel free to leave a question or interrupt me anytime. So I think Yon gave really good introduction to cryo-ET. So today I'm just going to talk about very basic overview of what the cryo-ET is. So like, I'm sorry. I should do it. So the main difference is that from single particle cryo-EM, cryo-ET is that we tilt the stage. So we take 2D projections of our object at different tilt angle using computational method. We back project those images to get to the 3D image of our specimen. So here's a simple diagram of that same movie I just showed you. So we tilt usually between plus and minus 60 degree, and with increment anywhere between 2 degrees to 3 degrees. Once you have those 2D images stacks, we call tilt series. We project it back and reconstruct what we call a tomogram. The tomogram is 3D view of your specimen. So here's an example of a tomogram. Specifically, we're looking at bacteria pathogen called Legionella pneumophila. There, you can already identify there are a few different features, such as cell membranes, a ribosome, or secretion machinery, like I labeled here. So this is going up and down through the Z slice of a tomogram. Like I mentioned, this is all in three-dimensional space. So we can do a lot of cool processing to make this into three-dimensional objects, and also do averaging, put it back into the native environments. We can also study how each macromolecular are interacting in a native environment. So for those of you who are new to cryo-ET or cryo-EM, or both of them, so here is like a simple difference between single particle cryo-EM and cryo-ET. In single particle cryo-EM, we collect movie series in one location, and after you draft correct it, you get one 2D image. Whereas in cryo-ET, like I mentioned, we tilt our specimen at different angle. So in one specific target, we get about 35 to 40 different stacks of movie images. And when we drift correct this image, instead of having one 2D projection, we are ended up with 35 different projection images. And these images are subject for what we call tilt series alignment. So we have to align these images one to the other so that we can have a smooth transition of one image to the other. And once you have reasonably well-aligned tilt series, this goes into tomogram reconstruction. Now you have what we call 3D tomogram. So those are the fundamental difference between single particle cryo-EM and cryo-ET. And there are many considerations when you collect this type of data, and one of them is how you're going to the data collection schemes. So in the early days, people experimented with different ways of collecting data. For example, unidirectional tilt is starting at one high angle to the next high angle, just nonstop data collection in one direction. Bidirectional tilt is sort of in the middle, where you start from one angle and then go halfway and then continue from the next half the tilt series. But what's sort of accepted in the community is that the best way to collect data is what we call dosimetric tilt skewing. So we start from almost 0 degree, and we collect left and right angle, plus and minus angle, sort of simultaneously. So you can distribute electron dose evenly on both extreme ends of your tilt series. So once you collect this type of tilt series, like I mentioned, we have to align the tilt series. This is because, of course, it'll be wonderful if the stage is entirely stable, so that as you tilt, you're perfectly collecting images on the same area. But in reality, that's not true. Because of this imperfection, type of image you have is like what I have illustrated on the left. So your images are sort of all over at different angles. So by going through a process called tilt series alignment, you can nicely align all the tilt images sort of at tilt axis. So how do you do this? One of the most popular way or traditional way of doing this is applying gold nanoparticles in your sample, like I highlighted here. And then using this gold particle as a fiducial, you can use software such as IMOD to align the location of these beads at every tilt images. And now there are more newer ways of doing this, and one of the most popular one in the field now is software called Arctomo. So this type of software aligns tilt images without any marker, and this is particularly useful. I'll get to it later. When you deal with biofilm mill samples, you cannot apply this gold particles inside the cell. So you'll have to find a way to align your data bark-free, and this software has been working very well in my lab, and I guess many other people's hands. There are mainly two ways of doing this, with fiducial and without fiducial. And another parameter that we have to consider when we collect this type of data is what is the range of our tilt series and what is our increment. So the range is, for example, here from minus to plus 70, minus 60 to plus 60. So because the way the microscope is built, we cannot collect data from minus 90 to plus 90 degree. This will be the most ideal scenario, but we just simply cannot do it. So we are mostly collecting data around this region between 60 to 50 degrees. And as you see in these images, because we do that, there is an artifact called missing wedge effect, that we're actually missing some information from the angles that we are not collecting the data. And when you compare between 2 degree increment and 5 degree increment, you can see that going with smaller increment angle gives you more complete information back. But all these things have to be sort of designed realistically, because if we set our increment too small, we have to take a lot more images on the same sample than you'll accumulate too much radiation damage, so your sample will end up burning. So you'll have to find a very fine balance between what gives you a reasonable image at a reasonable total dose. So I really love this type of diagram. Particularly, this diagram is from Thermo Fisher. Cryo-electron tomography is really unique, operates at a range that are very unique uh, in a way that this is perfect medium between traditional structural biology, such as single particle cryo-EM or X-ray crystallography, with what we consider cell biology, for example, light microscopy or SEM, conventional SEM, TEM. So before I go into the deep sort of list of examples where cryo-ET is used very usefully, I want to talk about a few key concepts here before we move on. So subtomogram averaging is sort of similar to particle alignment in single particle cryo-EM, but like the name suggests, we are using subtomogram, which is a small 3D volume of your interest that you want to extract from your tomogram. And once you have this objects identified and extracted, then you can align one another to a point that every particle sort of aligns in a single orientation that will give you improved resolution. And then this process can be done iteratively. So technically, it's very similar to single particle reconstruction, but then in this case, we're using 3D raw material versus 2D raw material. Of course, now there are softwares that uses 3D tomograms, but then refines the particle in 2D images. So those are sort of the latest technology. I'll get to in a few slides. One of the most challenging part of doing subtomogram averaging is particle picking, in my opinion. So we can directly pick particles just like single particle. But then imagine we're picking particles in three-dimensional space, not two-dimensional. So that adds a layer of complexity in terms of how well the softwares can pick the particles. So there have been, sometimes we still manually pick particles, but there are many software that does template matching type of particle picking, or machine learning type of algorithm where they learn positive and negative reference, and they can pick based on what they learn from the algorithm. And there are actually quite unique particle picking approach that are quite unique to cryo-ET. One of them is geometry-based particle picking. So, for example, this specimen here is HIV GAC protein. It forms this nice spherical shape. So what you can do is you can fit a 3D sphere in the middle of these type of virus particles, and then you can oversample along the surface of the shape you are fitting into. So this type of approach has been very successful, particularly in John Brick's groups when they study this HIV GAC proteins. Sort of improved version of this geometric-based picking is segmentation-based picking. So segmentation is the way to annotate the feature of interest in 3D objects. So in this case, I segmented the membrane of the bacteria, and then we oversample on the surface of this segmented volume. So you can actually oversample every object that are on the surface of, in this case, bacteria or any particle of your interest. So speaking of segmentation, segmentation is really the integral part of cryo-ET, because a lot of times we are dealing with cellular environment. We want to nicely annotate the important feature that can clearly show to the reader what we are looking at. So this is example from one particular software called Membrane. So this software is really good at picking up membranes in the cell. And as you can see, you can resolve this beautiful stack of membranes very easily. So really beyond the beautiful representation of your tomogram, you can actually use this segmentation to analyze your data set. So we have this software, particular software called morphometrics softwares. You can use this software to measure, for example, intermembrane spacing or the curvedness of your membrane. I think this is very important because when you look inside cellular environment, it's really a jungle. There are just so many features that looks so different from one another. This type of analysis allows you to really decode the interesting biology that's hidden inside this complicated tomograph. Like what Young sort of previewed, so I think the old notion is that people are thinking that cryo-ET is really for low resolution averaging work, but that has been, the idea has been changing over time. Of course, thanks to development of new algorithm to reconstruct averages and also to prepare a sample. So most of the high resolution examples we have in the literature comes from particles that are usually abundant in a sample, such as ribosome, and in the case of virus, this is spike protein from SARS-CoV-2. So my personal belief is that if you are after a biological system or a macromolecule that are abundant in your tomogram, I really think getting a high resolution these days is not very difficult. So if you are going for a high resolution tomograph averaging type of project, you should probably optimize your sample in such a way that you can have abundance of particles that you can average from. So combining all these useful tools, tomogram averaging and segmentation, you can do sort of very interesting analysis, such as in this example here, you can average many different types of macromolecules and put it back into where they came from and do analysis of how these different macromolecules are interacting with one another inside the cell and also within the organellar environment. So in cryo-ET, sample thickness is really the key limitation here. It's all because of this concept of mean free path. So for 3 kV TEM microscope, really, if you want to have high resolution imaging, you need to limit your sample thickness below 280 nanometer. And that is why historically cryo-ET has been used on specimens such as bacteria, virus, purified organelles, and in some cases, very thin areas of mammalian cell. So, just as an example, cryo-ET been very valuable in studying the structure of bacterial secretion system. These are type of nanomachineries that cannot be purified as an entire complex. So the only way to really study the structure of this type of complex is really looking at in the cell in the most native environment. So we have two examples from one is type 3 secretion system from Salmonella, and also efflux pump. I believe this is from E. coli. And also systems such as bacterial flagellum motors. So these are the nanomachinery that propels bacteria to move forward and backward. Again, this type of large complexes have never been purified as a whole. So really, cryo-ET uniquely suit in this regime of sample size and type of complexes. And of course, aside from average interesting nanomachinery from bacteria, cryo-ET can be used to look at simply interesting bacteriology. So one example I have on the left is these are sort of what we call vampire bacteria. So you can imagine this brown membrane and then the bacteria that's inside. So these are two different bacteria, but one invades the other. So they sort of hide and live a parasitic life inside another bacteria. So this is very interesting biology that cryo-ET was very uniquely suited to study what is going on. And very recently, we have this also paper where talking about a bacteria and how they have a non-membrane associated channel that transports things outside of the bacteria. So also, this is another example of interesting biology or bacteriology you can go after using this technology. Like I mentioned, virus is another popular specimen of cryo-ET. So in the study of HIV spike protein, cryo-ET have been used quite routinely. So the left is the first ever structure of HIV spike on the surface of virus. And of course, over time, with a better microscope and software, we improve the resolution of the same complex on the surface of virus. And even moving further, now we can study how this viral spike interacts with host receptors that are membrane bound. So this is a very recent paper from Yale. And of course, I think a lot of people know Tom Brick's group did really this groundbreaking work on understanding how HIV capsid undergo from immature to mature state, and they resolved this capsid lattice in a very high resolution. And this is quite a popular example of high resolution, so tomograph average. And also, cryo-ET was particularly powerful during the pandemic. People use cryo-ET to understand the molecular architecture of SARS-CoV-2. And also sort of looking at it in a most native context allowed us to understand how flexible the viral spike is relative to the viral surface, and also how some spike proteins have partially open receptor binding domain. I think this was a very interesting point of debate when people are comparing purified spike versus the native spike, and whether this partial opening of the receptor binding domain is true on the surface of real virus, or is it a artifact of purifying this complex. So cryo-ET was really powerful in the pandemic in understanding the molecular nature of SARS-CoV-2. And like I mentioned, thin edges of mammalian cells can also be imaged using cryo-ET. One example is how bacteria such as Salmonella interact with the host cell. This is HeLa cell at the edges using this needle type of apparatus. And another popular specimen is neurons. Neurons tend to grow very thin on the surface of cryo-EM grid. So people use this specimen to study how the communication between different neuronal cells and the synapses. So what I showed you so far is specimens that are less than, let's say, 280 nanometers. So what if my specimen is thicker than that? So we have this technology called cryo-focused ion milling. So we're using ion beam to cut top and bottom of the specimen, in this case, a mammalian cell, leaving a small section where you can do a cryo-ET imaging on it. And this is just one example of such specimen that I generated here in script. This is just a biofilm of a bacteria. So now the question is, how do I know where I mill? So you can only mill a window of about 10 micron, which is very small compared to the entire cell. So we use technology called cryo-correlative light and electron microscopy, in short, cryo-CLEM. So the idea is very simple. If your cell has a target that fluoresces, we can put our frozen specimen into these specialized microscopes and excuse me, identify where the target of interest is using the fluorescent signal. And using this information, you can go ahead and do the milling so that you can retain the target of interest within your lamella. So in terms of the workflow, now there are sort of two major ways of doing this. So the more conventional way is, once you have a sample prepared, it goes into the dedicated cryo-CLEM microscope. Once you get fluorescent images, the sample and the image is imported into cryo-EM milling, and then you do a correlation to mill the sample, and it goes into the TEM. So another workflow is that there are some companies that makes the camera that can go directly into the film milling machine. So you can, once you have your sample prepared, it can go directly into the film milling microscope, do the fluorescent imaging and correlation and milling all at the same time, and then that sample goes into the microscope. The way I presented it makes the first pipeline, probably more laborious and long. It's compared to the second workflow, it is partly true, but then each method actually have its own advantages and disadvantages. So here's one example of cryo-clamp. So in this paper, they label their lipid droplet with fluorescent tag. So in A, you can sort of identify the region of interest. B is to correlate the fluorescent images with SEM image. Once you know where you want to mill, then you can directly mill that area of interest. And when you overlay that fluorescent signal with the SEM image, you can nicely correlate both of the signals on top of each other. Using this type of pipeline, I was able to find, in this case, salmonella bacteria that's inside primary macrophage cells that are specifically interacting with mitochondria. So once I identified this target, I correlated and mill using cryo-focused heavy milling, and then take it to the cryo-ET so you can precisely find the target of interest that you are after. And a lot of time, so this type of analysis works best if you really know what you're looking at. For example, in this case, I'm looking for a bacteria. So in my TEM images, I'll naturally look for what looks like a bacteria. So it's actually very easy to find those. And same for cases like this, where if you're looking for a large object like a droplet, you can probably correlate your signal very precisely. But what if you're after macromolecule that's too small to be identified by your eyes? So a lot of labs have been developing new tools or using what's known to sort of identify identity of a small target in TEM images. So in the first case, so antibody, particularly IgG, is actually big enough to be identified in cryo-ET images. So in this particular example, we put a tag at the tip of this needle complex, and we added IgG that's specifically probing for that protein, and you can clearly see that this antibody density sort of decorates the protein of interest. And there was a group where people used DNA origami, which looks very unique because there's nothing that looks like DNA or origami inside a cell. So they use this type of tag to identify where is the target of your interest. And also there's a group that uses genetically encoded tag. So these are called GEMs. So you can tag this just like how you would tag GFP to your protein of your interest. It will form a small spherical cage next to your target of your interest. So you can use this tag visually to identify where your protein of interest is. So this is my last slide. So what is there for cryo-ET? So where is this going? So I really believe that the future of cryo-ET is really in interdisciplinary research and really more biology. So now there are a lot of tools such as AlphaFold, or in cases there are available crystal structure or EM structure that you can map your sort of lower resolution cryo-ET map into, but then having more information such as fluorescent images or live imaging really complements the cryo-ET data, which is simply black and white. So cryo-ET is really great technology, but it does not have information such as dynamics. So having complementary technologies such as light imaging or any other secondary experiment is really crucial in validating your cryo-ET data. So really putting everything together is important. And I mentioned here biology. It's because this is really my personal experience. And when I talk to many people that starts cryo-ET or cryo milling, everyone shares the same idea that once you get your image, it's beautiful, but oftentimes we don't know what's going on. So going back to biology, really characterizing your image using multiple different methods, like even going back to genetics, imaging, and all kinds of biophysical assays are really fundamental to doing good cryo-ET work. So I would say the key to cryo-ET and then the future of cryo-ET is really on more biology. With that, that's my last slide. Thank you.
Thank you, Raphael, for such a wonderful overview of cryo-electron tomography. I think here's one question in the chat. So what are the current challenges of cryo-CLAM? Are there any, or does it work fairly well?
So biggest challenge of cryo-CLAM is correlation. So correlation in terms of putting your fluorescent images, precisely overlay them in X, Y, and Z coordinate so that you can really capture the event of your interest within the 200 nanometer thickness of lamella. So I would say correlation is the biggest hurdle in cryo-CLAM.
Okay, here's another one. So who is developing the software used to build the images in this field? Private companies or public research groups?
Can you repeat the question again? I'm sorry.
Who are developing the softwares for cryo-ET? Private companies or public research groups? I think mainly it's from the public research groups, right?
Yeah, it's mainly from public research group, and cryo-ET community is very generous. So to my knowledge, most of the software are open source. So it helps the whole community to build the technology together.
So the next question is for the missing wedge effect, why can't we tilt from positive 90 degree to minus 90 degree all the way up?
Oh, it's because if you imagine the microscope, the beam is coming. I mean, if you look at microscope, it's sort of tall, right? And then the beam is coming from top to the bottom. And your specimen cannot physically be imaged at the exactly same angle where the beam is coming down. So that's why it cannot be imaged 90 degrees to another 90.
Yes, it's just like a book, so it's very thin, but when you lift it over to 90 degree, it's much, much thicker. And also the TM grid is made of metal, so it will block most of the signal at all.
Right.
Okay, so I think that's all the questions I can read in the chat window. Probably now we can start our morning session, discussion session. So if anyone has any questions, please unmute yourself and turn on your camera so you can ask questions to either Yun or Raphael. So maybe I can ask one question. So just now related to the question about the fluorescence probe. I think you mentioned about the XY position. I think the fluorescence probe really helps to identify it horizontally. But since we can see the probe along all the Z directions, right? Fluorescence signal, even it's at the bottom of the cell, I can still see it from the top. So in that way, it cannot really help us to identify in the Z direction, right? Oh.
Yeah. So a lot of these cryo-EM images are Z stacks. So it has, like you mentioned, all the Z information. But then correlating that image into your SEM image is actually very challenging. So there are ways to do this. We use small fluorescent beads where you can align fluorescent signal and SEM images into. So it's not, I mean, it's pretty good these days, but it's not perfect. And it's not easy to get perfect correlation. So that's the challenge there. Yeah, it is possible.
So while we are waiting, maybe I can reshare. How do you deal with protein heterogeneity in cryo-electron tomography?
Oh yeah, this is dealt in a very similar way as single particle cryo-EM. So we also do classifications, and also there's new programs where you can study heterogeneity in the motion, like cryo-ET, the same way single particle is dealing with.
Okay, thank you. Miguel, feel free to unmute yourself.
Okay, I tried. Can you hear me?
Yes.
Okay. Yeah, probably I'm not asking anything new, but instead of rotating the sample, has anyone tried to cut it in different directions so that you always have the same thickness? I know it sounds like it's not like cutting a cake. I know it's a lot more difficult. Just probably a silly question, if you will.
I believe that has never been done, but your idea is sort of cutting the sample at different angle rather than tilting the stage, right? I guess you can think of it this way. It's more economical to have one sample tilted rather than having different angle. And also, if you cut sample at different angle, you're actually looking at different specimen, like different cell or different virus, different bacteria. So it's difficult to get all this full range of angles in your
Sure.
images.
But just thinking of the idea of, okay, you just do it with many of them, and they are all cut in different slices, right? So one you're looking here, but the other one you cut like that. So you always have the same thickness. But of course, again, you're going to need millions likewise, right?
Interesting idea.
No, I think probably it's less in the theory. If we do the regular cryo-electron tomography, we want to look at the same target, but tilt the sample in order to get different views. So in this sense, so whether we cut it this way or the other way, we have to tilt it. Okay, here's another question in the chat. Which tool do you use for cryo-electron tomography segmentation? I think you mentioned about MR, the EM tool, right?
Right. There are actually a lot of options out there. One of my favorite is EM2 segmentation. It's really good. And also Dragonfly is another free software you can use. It's really good. And the software I mentioned in my talk called Membrane Seg is particularly good at segmenting membranes automatically. So that's also very powerful tool.
And also in the afternoon session, let me see. Actually, I think tomorrow, so in Muyan Chen's token, he may mention some of it using EM2. It's not purely the segmentation, but kind of related. Okay, so I think while we are waiting for more questions, so I would talk about today's agenda. So this afternoon, the course will resume at 1 p.m. Eastern Time. So Dr. Jianfeng Lin will talk about sample preparation using cryo-FIB. Then Dr. Phil Guo will talk about the cryo-ET data collection. And also in the email, we were asked where the lecture notes will be. So the lecture notes, after we receive it, we will post it on the Cryo-EM course website. And also, since we are posting the videos on YouTube channel, so we'll put a link there too. So all the speakers, please upload your presentation. I think the link was sent a few weeks ago. Okay, so I think now we got more questions. First one is for you. Did you also have to identify different locations for meeting, as mentioned by Dr. Parks, using fluorescence method?
Not really. For our current experiment, I think the advantage probably is the throughput and brute force. Just collect everything in the cell. So build up the abundance that way. But I do agree, using some, as I mentioned, some clamp-based approaches to concentrate the collection area to those places you know your molecules are concentrated, probably a better approach. It's really, everything is about throughput. If people can develop a very robust and convenient way to do clamp type of research, will be fantastic.
Thank you. So the next question is for Raphael. So what type of fluorescence imaging is used in cryo-clamp? Is it just a simple dye-based fluorescence method or fancier ones like two-photon or second harmonic generation?
Oh, this is just very simple dye-based fluorescence. You can imagine you can do the same thing with your other fluorescent light imaging experiment. So cryo-clamp microscope is actually nothing special, but microscope with cryo stage to keep your sample cold, and using a special objective that are also not sensitive to low temperature. So you can generate this cryo-clamp sample just like you would generate any type of fluorescent experiment sample.
Okay, so do you care about the thickness and the quality in the case of sample of cells that undergo film milling, or you just need the cell to be well frozen?
Yeah, so for example, if your mammalian cell is too thick, it will not freeze quick enough that it will actually accumulate, crystallize in your sample. So still, even though you're going to mill the sample, it's important that you have a sample that's relatively thin. And if that's not possible, you can also add cryoprotectants such as 10% glycerol to prevent the formation of crystalline ice, so you can get around this sample thickness and freezing issue. Yeah, but in general, even though you're going to build a sample, you want your sample to be thin.
Yeah, I can add a little on that to that. It's actually, we have similar experience. Even if your sample become too thick, it's very hard to cut through by the film milling equipment. The worst scenario is actually, these are very high power ion beams. They can cut through your grid bar, but there it's actually going to be very hard to cut through. A lot of time you end up lamella that's actually not fully processed, especially the polishing part can be very difficult if you're trying to cut the grid bars.
And I think it's mainly depends on the sample. Usually for suspended cells, we use the VitroBot to freeze it up to 10 micron commonly. But if the sample is much thicker, so I think at LVMS, we recently installed a high pressure freezer, so that can freeze sample up to 300 micron. But for light one, as you said, it's not possible to thin it using regular way. We have to do cryo lift out or use a cryo ultramicrotome to thin it. So I think that cryo-ET is really different from the single particle data analysis. The sample varies so much.
Yes.
Yeah.
Okay, I think there are no more questions. Probably we can stop here for the morning session. So thanks Yun and Raphael for such a wonderful presentation, and thanks everyone for attending and with all of the questions.